This is preparation for the second half of lecture 18. I wanted to do it separately because it was getting a bit too long. I will introduce the zebrafish. We aren't going to spend too much time talking about it, but there are places where it's very similar to the frog development, and a few places where it's a little bit different. So pay attention to those sort of similar, different um, um, aspects. And when we talk in later chapters about frog development, we will probably be making a comparison with zebrafish and looking at the molecules, because we will find that for certain processes, the molecules are practically the same. So they're very similar organisms in terms of their development. And so the whole life cycle is to the adult stage is about 3 months. So it's a relatively quick process. And only 2 days after, well, 2 and a bit days after fertilization, you have a little free swimming baby fish. It still has, let's just get the pen going. There's still a yolk sac there, so it's still able to, it develops a little bit further until it forms a complete fish body, and then grows to mature to the adult stage. About 30 minutes after fertilization, cleavage starts. Now the fish embryo, the little cytoplasm at the top of that first that first cell, which is the egg, the little clear bit at the top is where the nucleus is and where all the action takes place. The material on the other side is actually almost entirely yolk, not 100%. There are some cells in this region, but the vast majority of what we're going to be looking at is up here. So the cleavage stages in those first few hours, they take place in almost like a little cloud floating on the surface of the yolk. After about 4 hours, we have the blastoderm stage. It's like a blastula, right? But it's called, oops, sorry about that. And so, but still notice that like the embryo is all completely on the top. And some of these cells, we'll look at this later on, but some of the cells are kind of continuous with the yolk there. And the one thing that's different at this stage, and we'll see more about that later, is there is no blastocele cavity. So most embryos, even the frog with a huge amount of yolk and so on, there is an actual cavity, and we aren't going to see that. So the sphere stage is basically the blastoderm stage, and then the shield stage is when the cells, they have to grow down and around, and the idea is you have to enclose that yolk the way it is here, so that it's inside, and then the little embryo is kind of arranged outside of it. And so there's a bunch of cell movements that we're going to talk about. And so the gastrulation process and epibole, which is the way it's described, how the cells move down and around and cover the embryo. And it's probably not very clear here, but there's cell movements. So some of the cells make their way around, but others are actually moving inside, and a gut is forming in here, and anterior posterior. And then when we get to later stages, there are actually movements from all the way around the embryo. Remember, it's a ball. I'm gesturing in the air. That's not very helpful. The cells kind of move sort of towards the dorsal midline, and they generate this fish shape here. So at the beginning of the embryo, the cells are moving down and around, but then you get this sort of big rearrangement with migration, as well as some convergence to create the fish shape. And then we probably won't be talking about the development of somates, but just notice the major organs start to become visible pretty early on. It's only a day after fertilization. You can see the beginning of the eye. This is clearly the head and the tail. There are these somites, these segmentally repeated blocks of mesoderm that kind of pattern the body, including influencing the neural tube and that sort of thing, as well as the notochord, which is doing that job as well. So cleavage is confined in the zebrafish to the region above the yolk, and so you end up with these clusters of cells that are right on the top surface. And so, you know, this shows you, this isn't really the sphere stage, but if you make this more cells, thousands of cells that are smaller, notice how you have one cell and then you have two cells and the cells get smaller. There's not much growth at this stage. But that sphere stage is when you have about 1,000 cells or so floating above the yolk. So this is a schematic of excuse me, of the blastoderm. And so you could, if you took a slice through that blastoderm embryo, you would notice that there is a fate map possible. So if you did some cell marking and following, you would discover that in the top region is all the ectoderm, and below that is mesoderm, and then below that is a mixture of mesoendoderm, so mesoendoderm, mesoderm, endoderm. In the sea urchin, we refer to it as, I think, endomesoderm. Okay, so that's the same thing. It's cells with a mixture of fate, and there's also this is the yolk syncytial layer. There are some, you know, some of the cells, they don't finish dividing because it's just hard to get the cell membrane through that yolk. So you have at the bottom here, syncytial it refers to the fact that there's a lot of there's nuclei in here, and some of the you know, endoderm cells here are continuous with the yolk. Okay, and so the epibole starts right after this stage, and we start seeing the movement of these cells. And so there's a series of movements that we'll talk about. But the idea is the movement of these cells as well as the changing of shape. So you can see that the top layer, the cells are flatter and wider than the other cells. And the region at the edge of the actual embryo part, as it's moving down, it's referred to as the germ ring. So if you were looking at this as a ball, and you would be able to see sort of this ring around here. Remember, this is sliced in half. That's referred to as the germ ring, and that's the embryo proper moving down and around. Okay, back to the pen. And so as gastrulation begins, you can actually see some sort of puckering in this region. So there's a physical appearance that you can see. And some of the cells are going to continue to extend down and around. Those are going to be the ectodermal cells, but the mesoderm cells and the endomesoderm or mesendoderm start to curl around, and they start migrating back inside. This is called involution, because they move as sheets, and they move kind of around and then inwards. And then this is where the convergence and extension takes place. The cells are migrating. This is to form that fish body that I was telling you about. And so the cells are both migrating and then kind of intercalating with each other, doing what we would call convergent extension, but this includes as well these sort of migration, so that you end up with, on the ball of yolk, along the dorsal side, you're going to end up with this this fish body, because all the cells have, you know, moved around, and they're now forming a full embryo with all its proper structures and so on. A recognizable fish shape. Okay, so these are some of the types of movements that we're going to look at. So epibole is when the cells, the ones remaining on the outside, they're flattening and widening, and so that causes them to move, to cover a larger space. But also in the region immediately underneath, there are different layers of cells, and these are basically mixing together, kind of taking turns sorting themselves out, but making this is a thinner but wider ring. The idea being that instead of that ball of cells right at the top of the yolk that's narrowing down, flattening out by both of these processes, epibole and this kind of intercalation. It's when rings of cells kind of connect to each other. That's what the radial is referring to, and they intersperse with each other and form this sort of narrower strip. And then the involution, which is actually the cells moving around this way, and this is the movement of sheets of cells. And so I've already sort of talked about this. I guess I meant to talk about them separately. But this is basically what I just finished saying, how the cells move down and around to enclose the embryo. Now, it's possible, epibole, it's not necessarily that the cells are changing shape kind of their own accord. So if you were to imagine, stop that. There we are. So here is our upper layer of cells, and then here's some layers down below. And so as this radial intercalation is happening, which I've just made very simple by just making it a single row of cells. But if these guys start losing sort of their strong connections to each other and then kind of going in between, taking turns, as it were, like this to create a narrower but longer process. If the cells on top still maintain their connections to the red cells, as we're squeezing the purple cells between them, that's going to cause a kind of stress that just, and assuming also that these guys are strongly connected to each other, sort of side by side, that's going to pull them sort of flatter and wider. So this isn't necessarily an active process on the parts of these cells. It may be a consequence of the intercalation that's going on below them at the lower level, but because they are maintaining connections to the one layer of cells, and now we're squeezing additional cells in between, right? So that's kind of the idea about that. And then gastrulation by involution, this is when you have different layers of cells that are moving around and going inside inside the embryo. And the key is the idea is that the cells are moving as sheets or rows. So if you imagine you've got like all these different sets of cells, and they're approximately in rows, and they have these strong connections to each other side by side. And in the epibole example, we had these sort of strong connections between the upper layers and the lower layers and so on as well. But if, for example, we lose some of those connections, so you just have to rearrange your cytoskeleton and some of the proteins a little bit, and now the connections between each of the layers is less or gone. Maybe not zero, but it's a lot less. And there's still strong connections between the cells in the row, and so that allows one row of cells, to move in and around and inside the embryo, while leaving some of these other rows of cells on the outside, and they're going to actually complete the surrounding of the yolk. So I find sort of simplified diagrams like this can be helpful for understanding kind of what's happening on a larger scale. And then finally, convergent extension, where you've got, this is convergent extension where cells from two different directions are moving in and mixing together. And what's happening, again, think of this as a, this is a whole embryo, and so the cells are coming up from one side, they're coming up from the other side, and they're all kind of moving into this sort of dorsal midline of the embryo. And so you're going to have, like when we look here, there are some cells over the yolk here, but there's very few of them. The vast majority of the cells moved up to the dorsal side and on the other side as well to create, this embryo. So we went from this sort of ball of cells right at the top of the yolk to this really fish-like structure that is enclosing the yolk. So hopefully that's clear. These are really interesting. We will probably look at examples of these kinds of movements again. So, I mean, we've talked about them a couple of times already, which is one of the reasons why I'm mentioning them again. Okay, so this is just a summary of those movements and what the reason is for it. So the epiboly and the radial intercalation is to basically cover up the yolk. The yolk needs to be inside because as the embryo develops, it's going to be feeding off of the yolk. And then involution is basically the process of gastrulation, and that is the endoderm and the mesoderm needs to be moved to the interior so the endoderm can form the gut, the mesoderm can form notochord, the somites, and then eventually bones and muscles and internal organs and all that sort of thing. And then and that leaves the ectoderm on the outside, except for the neural tube, which gets internalized, and that gives you your epidermis and your nervous system. And then convergent extension to change that ball into an actual fish shape to get that more elongated form. Okay, so that's sort of all I really wanted to do. I wanted to introduce you to the fish and just some basics of its development. We aren't going to focus a lot on it, but there are some interesting observations, and so now you'll have some idea of where those fall in when we talk about them in a later chapter. Next, we're going to look at chick, and we might introduce mouse a little bit. Remember, this is just an introduction to the organism, so it's going to be pretty general. We'll look at details later with other chapters.